Thank you for
Okay,
joining
great.
us.
Yeah, no, thank you for inviting me. Yeah, and for like the really lovely introduction. As Helena mentioned, I'm Grace. I am a postdoc in my second year right now at the Harvard School of Engineering. I work with Hans-Peter Pfister, and my talk today is going to be about some research that I did before joining Harvard, but also a little bit of work that I did while I was here. And the large theme is going to be about building explainable AI tools for biomedical applications. Okay, so cool. All right. So when I talk about AI for biomedical applications, what am I referring to specifically, right? So I'm sure many of you have probably seen at this point, right, articles like on in newspapers and on social media about chatbots and LLMs that are being used for healthcare applications, and definitely also a lot of anecdotal evidence about how it goes wrong. I just want to say really quickly that this is not what I will be talking about for a large part of this talk today. And in fact, what I really care about is a different type of AI model. So I think AI has existed for a long time, right? Long before chatbots existed. And I think AI for biomedical healthcare applications has existed for almost just as long. And I think they've done a lot of really wonderful things for research in this field, right? So I think one of the most famous examples would be AlphaFold, which helps us predict the structure of proteins from the sequence of amino acids. And it's really helped us understand and develop therapies for diseases like sickle cell anemia. But of course, right, it doesn't stop there, right? I did a really quick search when I was putting this talk together on recent publications in the biomedical field. So I was looking at the UK Biobank data set, which was one that I've used a lot, right? And it really didn't take me very long before I found these examples about recent publications that are really AI models built on the data that was collected. And of course, you might say, well, some of these articles don't look like they're about AI, but then if you really start reading through them, you'll see that so much of it was really built on these old AI models from years ago, right? This is like 10 years ago now. Yeah, that helps us, segment and identify like white matter hyperintensities in the brain. Right. And then within the United States, if you live here right now, you'll see that like many of the AI-enabled medical devices, a large amount of them are like for radiology. So like there's just a lot of applications of AI in biomedical fields that is not really like anything about like, chatbots or LLMs, right? And this is really the kind of AI that I'm referring to in this talk. Okay, so then you might say, well, if AI for biomedical applications has existed for such a long time, and it's like and it's got like this proven track record, why do we need to explain it? And there are a few reasons, right? The most important one is that, sometimes AI models make mistakes, right? It's not 100% accurate, and there are always errors that are going to crop up. We need to be able to know and identify when it happens. But then I think also more importantly, right, we know that AI models can be biased. If the data that it's trained on has stereotypes, which, humans, we have stereotypes. I think that's unavoidable. And so if the data that we create has stereotypes and the models learn these stereotypes, they're going to replicate these biases. So we really need to be able to audit models and understand what they're doing. And then finally, and maybe most importantly, right, it's like, when these AI models, like, when they have flaws, right, who is responsible for them? Is it fair for us to go to, a healthcare expert, right, and say that, you are responsible for the outcomes of this model that you did not build, did not help train, and maybe do not even understand, right? I don't think that's a fair thing to say. And so I think there's a real need for us to more proactively involve biomedical experts, and by which I mean like doctors and scientists, right, in the development of AI models. And we need to create tools that help them understand what these models are doing. Okay, so this is the large motivation for a lot of my work, which is really about designing for the domain. And in this case, the domain is the biomedical domain, right? And as it turns out, this is actually really hard because within each domain within each expertise, right? The experts, they have their own prior knowledge, and they also have their own priorities, right? They don't have an infinite amount of time that they can spend like trying to understand a new like model that we put in front of them. And and then of course, there are other constraints such as, what tools do they have to work with? And then also like the fact that they need to communicate with other stakeholders. A doctor needs to talk to their patients, a researcher needs to talk to other researchers in the team. Right. So my talk today is going to focus on these two papers from recent years that really I would consider them case studies into how at least I in my work have looked at designing explainable AI tools for the biomedical domain. So the first of which is about developing counterfactual imaging techniques for magnetic resonance imaging, MRIs. And this was presented at FACT in 2023. And then more recently, I am going to present some work about contextualizing embedding spaces, right, in relation to the physical imaging data. So I'm just going to like dive into them right now. So the first paper is about creating counterfactual explanations of cardiovascular image classification models, right? And this was work that I did while I was still a PhD student at Georgia Tech. And you will see that, in the author list that I really worked very closely with like these two doctors from the Cleveland Clinic. Okay, so to just set the scene a little bit, right? What are MRI classification models? So here on screen, what you see is a cardiac MRI image from a patient, right? So this is someone who has gone to the doctor and gotten their heart imaged. And so what might happen is that we might train an AI model that looks at this image and then predicts the likelihood of this patient having some kind of disease, such as hypertension. And then so the model might, look at this image and then output a prediction that says 0, no, I don't think the patient has hypertension. So I think that's all well and good, but what we want to explain here is really why did the model create this output? And for a doctor, I think what the doctor will really care about is, does this prediction align with what the doctor themselves observe and what the doctor themselves would predict? Okay, so what do the experts want? So this is what we've found out through talking to our collaborators at the Cleveland Clinic, right? So they want to understand, which are the biological features that influence the image, the model's predictions based on in the image, right? And then what is it about those features that matter? So is it the color? Is it the shape? Is it the size? It could be any number of features that matter to the model's prediction. And then the most important thing is, does the model's behavior match established clinical knowledge? So for example, right, okay, sorry. I was blocked a little bit on the slide. Okay, so for example, in this particular MRI image that on screen right now, right, you will see that little part that I've highlighted with the little measurement rule, right? So that is the cardiac muscle. And so what we know clinically is that in patients with hypertension, this muscle is going to be a lot thicker, right? Because it needs to exert more pressure to counteract the pressure in the blood vessels. So when a doctor is inspecting an AI model, the doctor would probably want the AI model to do something where, if the patient has a thicker heart muscle, the model is going to predict hypertension with a greater likelihood. Okay, cool. So the technique that we used in our work to explain these AI models is counterfactual explanations. So what are counterfactual explanations? Counterfactual explanations are minimal perturbations that's made to the data to change the model's predictions. And they are very useful because they are model agnostic and they do not assume that the user knows anything about the underlying model architecture. So in a very simple example, imagine we're not working with images, we're working only with tabular data, right? So we have this patient with certain characteristics or variables on the left, right? And then like this patient is predicted to not have hypertension. And then on the right, what we see is the minimal change that's made to the patient's variables to change the model's prediction such that the patient now has hypertension. And we can see that this was, because it's a minimal change, we can see at once that the only change that was made was really to variable 2. And it was like, to increase the value from say 0.5 to 3.5, right? And so this allows us to do some kind of reasoning where we can say that, oh, it is because of this one variable and this one value that was increased, therefore the model's prediction was changed, right? So this allows us to reason about the model's behavior in an almost causal manner. So this is very well and good and like very useful in tabular data sets. But the challenge here is really, what does it mean to make a minimal perturbation for image or video data? Because that's the kind of data that we're working with, right? MRIs are not, a single like row of values, right? MRIs are very, very rich data sets. So there are some ways we can think about doing this, right? We can, for example, say, well, what if we like perturb the fewest number of pixels in this image? Or what if we perturb each pixel by the smallest amount? Or what if we find the nearest neighbor in the data set, right? These are all different ways we can approach the idea of making a minimal perturbation. And I just want to say that this technique already exists, right? So I'm sure some of you may have seen a similar example where we start with an image on the left that's a benign tumor, and then we perturb every single pixel by a very, very small value, and then suddenly, oh my God, the model now thinks that this image is one of a malignant tumor. That's horrible, right? And wow, like the perturbations that were made, right? The little pixel changes that were made, like in this image in the center, right? Like it's so small, it's a minimal perturbation, surely it works, right? And sure, I think it works, right? It's what we call an adversarial attack. But then I would argue that if we show these two counterfactual examples to a doctor, I don't think a doctor or any other human for that matter will be able to make sense of what the model is doing. It doesn't tell us, like we won't be able to identify what the change is, and I don't think we'll be able to reason about what the model is doing. So yeah, so it's probably not very understandable to humans. So in addition to the minimal perturbation requirement, in our work, we said that, okay, if we want to develop counterfactuals to explain these MRI classification models, we need to have two additional requirements, which is human interpretability and plausibility. So the counterfactual needs to be something that a human can reasonably look at and understand. Okay, so what does this really look? So how are we doing this, right? So say we have these two MRIs, right? One is predicted to have hypertension and one's predicted to have no hypertension. They have different outputs, but we can't really reason about why they have different outputs because they are different in so many places. In comparison, we can look at this image on the right, which also has a different output, right? So it has hypertension as opposed to the image in the middle, which has no hypertension, right? And then we can see that these two images are identical almost everywhere, except for this segment in the center, which is really the cardiac segments. And so we can really like immediately say like, well, it's really the change in these segments that's like causing the model to change its output prediction. So the question then is really, how do we generate this image on the right? How do we generate this minimal change, right? And the way we do it is we make use of segmentation models that are incredibly accurate and have existed for a very long time. So what we do in a segmentation model is we take an MRI, and then from this MRI, we use a model to identify the segments of the heart, or it could be really any biological segments in this image that the doctor would feel is relevant, should be relevant to a particular prediction. So in this case, it's the cardiac segments, which is the right ventricle cavity, your left ventricle cavity, your left ventricle myocardium. And then what we can do is we can start with the target image. This is the image we want to explain. And then say this target image is predicted to have no hypertension, right? And then so we start with this image, we segment out its cardiac segments, and then we find a source image that has the opposite prediction. So the source image is predicted to have hypertension. And then in the source image, we segment out the exact same cardiac segments. And then we basically do a little cut and paste from the source into the target. So we create this recombined image, which is identical to the target in every location except for the replaced cardiac segments. And then if we then put this recombined image through the predictive model, and then the model says, well, okay, I've changed my mind, the prediction has now switched from no hypertension to hypertension. We can say two things, right? We can say that, okay, it was the cardiac segments that really affected the model's prediction. And we can say that this recombined image is a counterfactual of the target. Okay, but at this point, I really want to pause and just highlight that, we don't really just want to know that the heart is important for model predictions. Like there are 100 other different explanation methods that tell us that the heart is important, right? Like a saliency map will tell us that, okay, the heart is important. We want to be able to make an explanation with a little bit more nuance. So from this example, what we can see is, remember what I was saying about the size of the thickness of the myocardium muscle in the heart, right? So we can see that, ideally, what we want to see is that the image that was predicted to have hypertension has a thicker myocardium muscle, right? So a doctor should be able to look at this and say, yes, this aligns with what I know about hypertension in real patients. And right, and then I think, and then sorry, to just finish the train of thought, ideally through multiple rounds of this validation and auditing, right, the doctor can then build up their own judgment of whether the model is doing what they expect it to do in a clinical setting. Okay, cool. So this is the large idea behind how our counterfactual explanation works, right? And then I do also want to say that, we don't just want to put an idea out there. We kind of want to demonstrate that it works, and that and that people can use it more easily if they wanted to. So we also created an open source implementation of this framework, right, that fits into existing workflows and works with existing tools, and we hope has a relatively low barrier to entry. So the way we did it was we kind of created this little interactive visualization tool that lives within Jupyter Lab. So essentially what happens is that, and probably not a doctor, maybe a data analyst working with a doctor, right, who's already, analyzing data within a Python, using Python Jupyter Notebook, right, can also do a pip install of our tool and then kind of load it within the notebook environment and then kind of interactively create these counterfactual images. And so there are, just as a quick, video demo, right, there are a few things you can do. You can load your target MRI, and then you can select the segments that you want to replace, right? And then once you have selected these segments, you can then also select your source images and then say, well, right? So you can select your source images, and the source images can be one or many, right? And you can filter based on the population demographics. And the reason we want to filter by population demographics is because there are many confounding factors that affect how people's hearts look, right? So the heart of a child is not going to look like the heart of an adult. If your target is an adult, you don't want to select source images from children, because that just completely messes up the reasoning that you make. So we want to be able to filter by demographic, and then we can select our source images in this way. And then this will then help us create a bunch of recombined counterfactual images. So I do want to say at this point that I'm not going to talk about the evaluation in details due to time constraints, but we did do an evaluation both algorithmically and also with actual doctors as our participants in the paper. So you're welcome to look at that if you're interested. Okay, so I'm actually just going to segue now into the second paper that I'm presenting today, which is about spatially resolved embedding analysis with linked imaging data. And this is work that happened more recently at the Pfister lab. Okay, so this is, so in this particular project, what we're working with is multiplexed cancer imaging data. So what I mean by that, so for those of you who have not worked with this data set before, right? What happens is that, say, if someone has cancer, we might take a biopsy of the cancer tissue, and then we will have a specimen that looks something like this on the left, right? And then within this specimen, right, within this little thin slice of tissue, there are a large number of cells that exist here, right? And each cell expresses one of many biomarkers, such as DNA and proteins, right? So you can see each cell has this little white outline around it. And then what happens is that, what happens is that for each cell, we can express the cell as a vector of its biomarkers. So essentially, like each value represents a biomarker and how much of that biomarker the cell is expressing. And we can do that for basically every single cell that's in this tissue/image, right? And so then what happens is that for each biomarker, we can create like a kind of image of how much that biomarker is expressed within this tissue. And then we can layer all the biomarkers on top of one another. And then we have this like grand overview of this entire slice of cancer tissue and all the cells and all the biomarkers that's being expressed by all the cells, right? And in theory, what's supposed to happen is that this will help us, identify cancer types, describe cancer types, describe how they proliferate, and then potentially, help us understand how we can develop, drug therapies to target cancer cells. But in reality, because there's so much going on, right? There's so many biomarkers, so many cells, so many layers. It's actually just too much data, right? It's very difficult for anyone to make a sense of all this data all at once. So we really need tools to help us do this, right? And visualization happens to be a really powerful approach that we can take. So traditional analysis methods, right? So this is the type of visualization that already exists in the field, right? What they use is embedding spaces, right? So embedding spaces allow us to so remember each cell is expressing like a long vector of multiple biomarkers. So embedding spaces allow us to collapse these long vectors into 2 dimensions, and then we can plot it in a single 2D scatter plot visualization. And so what's happening here is that each point in this visualization is a cell, right? And what happens is that points that are closer together are cells with similar biomarker expressions, and points that are further away are cells with different biomarker expressions. And the hope is that, by analyzing, how cells are similar or different, we can understand what distinguishes tumor cells from healthy cells and how we might, create therapies to target them. But this approach has a significant drawback, which is that it discards a lot of the spatial and morphological context that was in the original tissue, right? So we lose information about things like cell shape, cell neighborhoods, and proximity, right? So for example, if I had a very simple question, like are cells that have similar biomarker expressions also close to one another in the tissue? I actually cannot answer that with this embedding space. So it's one of the really large drawbacks. And so in our work, what we want to do is we want to kind of enhance this embedding space to also include spatial data, right? So we call this the hybrid embedding space, and it takes the original image and combines it with the embedding space that we have created. So how do we do that? So remember that in the original image, it's made up of a lot of different cells. So once again, what we can do is we can take a segmentation model. Segmentation models are really useful, by the way. So we can take a segmentation model and we can cut out each cell from the original spatial image, which is the original tissue. And then what we can do is we will try to place each cell at their embedding positions. Sorry, can everyone still hear me? My sound just went out. Okay, awesome, thank you.
It's working.
Okay, perfect. My headphones are having issues recently. Okay, so what happens is that we are cutting out each cell and we place them at their embedding positions. And so long as they don't overlap with another cell, we will place them in that position, right? So for example, I take my first cell, and obviously there's nothing in the embedding space, so I can place it. I take my second cell, it doesn't overlap with the first cell. That's great, I place it. And then I take my third cell, and so at low resolutions, it overlaps with the 2 cells that are already there, so I don't place it there. But at a high resolution, if I zoom in really close, it doesn't overlap with anything, so then I can place it. And so what this then allows us to do is we can create this hybrid embedding space where each cell is positioned where it would be, and then we can zoom in from low resolution to high resolution. And as we zoom in, more and more cells appear so that they don't overlap with one another and we can see them all clearly. Okay, so now that we have this, hybrid embedding space, we want to conduct some analysis with it, right? We don't just want to create this embedding space. We want it to tell us something. So embedding spaces are really powerful because they allow us to cluster entities together. So this is why we use them in the first place, right? And so the real insight here in this work is that we consider each cluster to be a set, right? It's like a mathematical set with its set of items. And these clusters can really be determined in different ways, right? We can have clusters that are determined algorithmically, like, for example, clustering algorithms. They can also be determined manually, for example, by user selections, right? I can draw an outline around a group of cells, and then I can say this is, a certain type of cell. This is another type of cell. And then by treating each cluster as a set, right, the intuition is that we can then apply mathematical set operations to compare and describe how each cluster is similar or different. So what do I mean by that, right? So take these two clusters on the left. So these are manually selected clusters. I, circled them in my hybrid embedding space, right? So each cluster has a certain distribution of the various biomarkers. So oh, sorry. This is from a case study. So this was something that one of our collaborators actually did. So each cluster has a certain distribution of biomarkers. And so what we can do is we can say, like, in the places where the cells overlap, right? What are the biomarkers, where the clusters overlap? What are the biomarkers in those cells? In the places where the biomarkers do not, where the clusters do not overlap, what are the biomarkers in those cells, right? So we can basically just go in and apply all the set operations that are available. So things like union, things like intersect, things like difference. And then like to actually talk about the case study that that our collaborator walked us through, right? So the reason they selected these two clusters was because he had originally thought that this was a cluster of B cells, right? And then he was like, oh, this is a really, really large cluster. It's much larger than I anticipated, right? And then so what he did was he then created these two selections within this cluster. So he made one cluster into two clusters. And then he looked at it and he said, oh, actually it's only like based on the biomarker distributions, it's really only the cells in the first set that are B cells, right? And everything else was actually a T cell. And that kind of made more sense because we don't actually have that many B cells in the tissue. Okay, and then I do also want to highlight that we have other case studies in the paper. I know that I have motivated a lot of this work using the biomedical application domain, right? But if we really think about it, the type of data that we're working with, which is, large imaging data with many individual entities where each entity expresses, um, expresses a vector of different attributes, right? This is not limited to the biomedical field. It's not limited to tissues. It can also be applied to other fields, like, for example, astronomical imaging, right? And so this is also another case study that we discussed in our paper, which is, like, can we take this same approach and use it in a different domain, like the astronomical domain, and like get at some useful analysis in that way? And turns out that yes, it's sort of useful, but I won't go into the details here. And okay, so and then of course, like no no paper is complete without an actual tool. So we did build a tool out of this, right? And this is what it looks like. And like in the previous paper, this is a tool that lives within the Jupyter computational notebook environment. So what you can do is you can install it using a pip install, and then you can get it to work with a lot of the existing data sets and data analysis workflows that you may already have. Okay, so that is a really just quick walkthrough of the two papers that I wanted to present, but I do also have some closing thoughts that's maybe just a reflection of the experience of working on papers like these, right? So most immediately, I think within the biomedical field, there are some very interesting questions, right, about scaling up the dimensions. So in both the MRI paper and then the cancer tissue imaging paper, right? Like we're working with largely 2D images, but we know that this is not always the case in the biomedical field. So as technology improves, right, like a lot of the data is increasingly high dimensional, right? It's so what this might mean would be that, for example, MRIs are often videos and not static images, right? And then also for multiplex cancer imaging, right? It's like they are also not like strictly 2D. Like a lot of them are like working towards like recreating the 3D shape of the cells as well within a 3D, volumetric representation, right? And then, of course, more broadly, there's also a general interest in, tracking patients across, long time spans of weeks and months and years. So what this means is that, like, in terms of both like the types of data that we have and then the types of analysis that we want to do, right? It's like we're really scaling up the dimensions. And so this really means that, there's probably going to be like new AI models that help us do that. But then with that, it also comes with like new explanation tools and like new explanation like requirements, right? Like how we validate that the model is doing what we think it does. And, I'm sure as we all know, visualization in 3D and across time is a lot harder than in 2D. And then, taking another step back, right? Like, if we're not just looking at a single subfield of of like the large biomedical domain, right? If we're not looking just at, cancer or if we're not looking just at, cardiac MRIs, right? It's like there is a need for, when we design for experts to incorporate domain knowledge into existing interpretability techniques. But I personally don't think that the solution is, every time there's a new model or every time there's a need to explain a model, right? We go to a small group of experts and then we work with this small group of experts and then we create a bespoke tool that works for the small group of experts. I don't think this is a solution that scales very well. So I think there has to be generalizable lessons that translate hopefully across domains, right? I think I hinted at this a little bit when I mentioned the fact that, the SEAL paper, like cancer imaging is really so much like astronomical imaging, right? But then where else do, domains like overlap and, what does it mean, right? For us to like build tools that are somewhat generalizable across these various domains. And then my final thought, which is I think the one that I worry about a little, is that I think very often explaining the model is not the same as explaining the science, right? So if we go back to AlphaFold, which I briefly mentioned at the start of this talk, AlphaFold it's like a phenomenal piece of technology, right? It helps us predict protein structures. But then I think at the end of the day, we don't know anything about protein folding. And my sense is that even if we were to be able to pick apart AlphaFold, we still wouldn't know anything about protein folding. And that is such an important thing to know for science and for developing, drug therapies, right? And so I think the allure of explanations is that, very often we mistake hypothesis generation for knowledge work, and that's not always the same thing. And so I feel like at the end of the day, it's like how useful really are explanations, and should we maybe be spending more time on knowledge discovery rather than explanations? I think that's something that I think about a lot. Okay, cool. So thank you. That brings me to the end of my talk. I want to thank my co-authors. I know some of them are here today. And I also really want to thank Helena for organizing this and for the wonderful audience for coming to the talk. All right, I'm happy to take any questions.